Welcome, everyone, to the last day of our virtual meeting for the Innovations in the Science of Cannabis Conference, put on by the Michael G. DeGroote Center for Medicinal Cannabis Research. It's my great pleasure to welcome our fourth keynote of the conference. Dr. Robert LaPrairie is the GlaxoSmithKline CIHR Chair in Drug Discovery and Development at the University of Saskatchewan. He is the Director of Education for the Canadian Consortium for the Investigation of Cannabinoids, CCIC, and also a member of the Cannabinoid Research Initiative of Saskatchewan, CRIS. Dr. LaPrairie's research focuses on the endocannabinoid system, and in particular, a critical receptor subtype, the CB1 receptor, which is both activated by our own endogenous neurotransmitters, the endocannabinoid system, but also by compounds like THC and CBD. He's a leader in this area of the field and one of the brightest rising stars in basic research on the endocannabinoid system and cannabis research. So it's my great pleasure to welcome him to join us today. Please join me in welcoming Dr. Robert LaPrairie from the University of Saskatchewan. Thanks so much for being here.
Thanks so much, Dr. McKillop, for that very generous introduction. And thank you to yourself and the entire organizing committee for inviting me to speak. This is a pleasure. So my name is Robert LaPrairie, as was mentioned, and I put my Twitter handle down at the bottom there. If anyone wants to message me, I'm welcome to take questions that don't get asked or answered during this talk. Today, I want to start by acknowledging that I am present here in Saskatoon on Treaty 6 territory and the homeland of the Métis. And the city of Hamilton, where McMaster is situated, is on the traditional territories of the Erie, Huron-Wendat, Haudenosaunee, and Mississaugas people. The land is covered by the Dish with One Spoon Wampum Belt Covenant, which was an agreement between the Haudenosaunee and Anishinaabeg to share and care for the resources around the Great Lakes. We further acknowledge that this land is covered by the Between the Lakes Purchase of 1792 and between the Crown and Mississaugas of the credit of the First Nation. So we start by paying our respect to the First Nations and Métis ancestors of this place and with one another. Some conflicts of interest to kick things off. So research in my lab is sponsored by GSK. However, GSK doesn't get to decide what we do in the lab. They give generous monies, but they do not oversee any of the study design goals or outcomes. Some of the research materials in my lab are purchased from Lafrary Cannabis. They also don't have any input on study design. And I receive speaking honoraria from various groups, including the CCIC and the Saskatchewan Health Authority, as well as others. And I'm acting as a consultant into ongoing court cases. So this is what I'm going to be doing today. I'm going to start with a brief history of the cannabinoid system at the level of molecular pharmacology. And I'm going to introduce some concepts regarding the endocannabinoid system and cannabinoids as medicine from a molecular perspective. So some of it might be a little bit of repetition from what we've heard from the previous keynote speakers and others, but my focus is really going to be on the molecular side of things. Then I'm going to talk about the fun stuff that's going on in my lab. I've got work in programs looking at synthetic cannabinoid receptor agonists, also known as SPICE, as well as a program looking at positive allosteric modulators of the cannabinoid receptors. I'll close with a little bit on our phytocannabinoids work and look ahead to see what the future holds. Also, just going to give you guys a heads up, there's going to be a lot of chemistry in this talk. So just think back to all those glorious, beautiful memories you have of second-year organic chemistry in your undergrad and how great those were. So if we were in person, I would ask you guys by show of hands, who has received education as part of your formal training about the endocannabinoid system? And with the exception of this symposium this week, my guess or supposition would be that the majority of you have received zero or no education in this area. I got one raised hand, thank you. So these things are very important to our biology as human beings, and yet because of a lag in research associated with the illegality of cannabis, there's been a substantial lag in our learning and education on this. So I like to put up this graph just to highlight this. In this graph, this is just data that I pulled off of PubMed earlier this week. What we can see is the number of papers published on cannabinoids broadly over the years. And I've highlighted a couple of key hallmarks. So here we see the discovery of delta-9 THC by Dr. Raphael Mechoulam in 1964. The cloning of the type 1 cannabinoid receptor, or CB1, in the early 90s, followed by CB2 in the mid-90s. Crystal structures for the cannabinoid receptors not occurring until 2016, and then federal cannabis legalization in Canada for both recreational and medicinal purposes in 2018. So that's kind of the timeline if we look at cannabinoid research. But if we look and compare that to other fields, and I'm going to highlight the opioid research field here, we can see there's quite a big lag in differences. So the first characterization of morphine as a molecule actually happened in 1804, so it wouldn't even appear on this graph. The first paper describing opioid receptors came out in 1960, and the first characterization of the endogenous opioids was in 1975. So if we characterize and look between fields, we can see, we've got a lot of ground to gain in this field. So then let's start by just defining the cannabinoid system from a molecular perspective. So generally speaking, a cannabinoid is any compound or ligand that is structurally or functionally related to delta-9 THC, delta-9 THC being down here in the middle. Delta-9 THC, as we know, comes from the cannabis sativa plant. It is the thing in cannabis that is intoxicating and psychoactive, and it's a partial agonist of our type 1 and type 2 cannabinoid receptors. We also have endocannabinoids or endogenous cannabinoids. These are naturally produced by our body and include things like anandamide and 2-AG, and they're also partial agonists at CB1 and CB2. We have things like cannabidiol, cannabidiol being also a psychoactive ligand in that it acts in the brain, but it is a non-intoxicating constituent of cannabis. And then we have our many synthetic cannabinoids, which were originally produced as tool compounds to help researchers study the endocannabinoid system. But more recently, various synthetic cannabinoids have appeared on the illicit market as drugs of misuse. And here are two examples here, WIN and CP. Synthetic cannabinoids, unlike others, tend to be full agonists at CB1, which gives rise to a number of different receptor-related adverse effects. So back in the 1970s, people thought that cannabinoids were false steroids in that they acted on steroid receptors. It wasn't known at the time how cannabinoids worked at the molecular level. Later on, in order to try and explain how cannabinoids had widespread and disparate effects on the body, it was simply thought that cannabinoids as lipids alter membrane fluidity. And it wasn't until 1988 that researchers, including Lynn Howlett, shown here, were able to show that there was a receptor-dependent mechanism that mediated cannabinoid pharmacology. And they were able to show this for cyclic AMP modulation being a saturable effect that was temperature-dependent. Later, researchers would go on to show that there were specific regions here of the rat brain where you could have binding of radiolabeled THC to specific receptors. It was later determined that that receptor was the type 1 cannabinoid receptor, which is the major receptor that causes intoxicating effects when activated. Following on this work, we did see the first cloning of a cannabinoid receptor in 1990, and then in situ mapping of CB1 mRNA, which matched the radiolabeled THC. In 1992 and 1995, we saw the purification and characterization of endogenous cannabinoids, anandamide and 2-arachidonoglycerol. And then in 93, we got a second cannabinoid receptor, the type 2 cannabinoid receptor, which is predominantly expressed in immune modulatory tissues and has a role in inflammation. So here we have a diagrammatic image of the endocannabinoid system in the central nervous system. And I'm just going to walk through this to kind of give context for how cannabinoid receptors are functioning within our neurons. So here we have an excitatory neuron that's going to release glutamate. When glutamate is released, it crosses the synaptic cleft where it activates and depolarizes the postsynaptic neuron. When that occurs, that postsynaptic neuron is going to on-demand synthesize endocannabinoids like 2-arachidonoglycerol. Unlike most neurotransmitters, 2-AG is going to travel in a retrograde direction back to the presynaptic neuron where it acts on CB1, here shown as this purple snake. When CB1 is activated, it's going to reduce cyclic AMP levels, it's going to inhibit neurotransmitter release, and it'll conduct a few other changes in the cell, all to the goal of reducing neurotransmitter release from that presynaptic neuron. And this was originally shown in 2001 by Kreitzer and Rager in this paper in Neuron, which if you have students or if you yourself are a student and you'd like a really solid paper to help you understand e-phys experiments, this is a great paper to walk you through some really well-conducted and nuanced e-phys experiments. So the type 1 cannabinoid receptor is what I'm going to spend most of my time talking about. It is the most abundant G protein coupled receptor in the central nervous system. And its pattern of expression changes as a function of our development. Because it's heavily involved in the regulation of neurotransmitter release, it's also involved in synaptic pruning and synaptic architecture. So in this graphic, what I'm trying to show is how CB1 distribution changes as a function of the lifespan. In mid to late embryonic development, what we can see is that CB1 is everywhere. Dark green shows areas with a very high CB1, and light green shows moderate levels of CB1 expression. And it's thought that CB1 levels are high in the developing brain in order to facilitate selective synaptic pruning or synaptogenesis and help the brain establish the important connections. Later on in adolescence, we see the pattern of expression shift, and CB1 is highest in the prefrontal cortex relative to other regions. And in adolescence, that prefrontal cortex is still rapidly undergoing maturation and hasn't fully formed all of its adult connections yet. And then in adulthood, the pattern of CB1 expression shifts still. And as adults, we have CB1 throughout our brain, but in highest levels in the midbrain and the cerebellum. So as adults, where do we see CB1? Well, we really see CB1 in parts of the brain that we might anecdotally associate with the effects of THC on the body. So for example, we know that in some people, THC use causes a hunger or an appetite increase. This is because of CB1 expression in the hypothalamus, shown here. For some individuals, CB1 activation is associated with anxiolytic or anxiety-reducing effects. This is because of CB1's presence in the amygdala. CB1 is also heavily involved in motor regulatory pathways throughout the caudate nucleus and the putamen, as well as the cerebral cortex. And so we can think about these different regions as being regions of highest relative CB1 expression. CB1 is found in other parts of the body as well, including in the gut, where it's involved in slowing down contraction of the gut. And in contrast, we see CB2, the other cannabinoid receptor, predominantly expressed in areas of the immunomodulatory system, where it causes anti-inflammatory effects. So here's a summary of everything I just talked about. CB1's job really is to inhibit presynaptic neurotransmitter release. Through this, it regulates various functions in the body, including pain perception, mood, anxiety, reward, satiety, locomotion, and body temperature. CB2, again, primarily found on immune cells and is involved in suppressing the release of pro-inflammatory cytokines. So now I'm going to transition and talk briefly about cannabis and how the different cannabinoids in cannabis might influence the plant. And one thing that I like to say before I get started is when I give talks to public individuals outside of the research space, one of the pieces of feedback I get is that cannabis is natural and therefore it is safe. But human beings tend to be really good at finding things in nature which are not all that safe. The dose determines the poison. And here we have some examples of an opium poppy, a coca plant where we get cocaine from, a tobacco plant, and a mescaline cactus. So all great examples in nature. of things that at higher doses maybe aren't so healthy for us. Cannabis sativa has been used by human beings medicinally and recreationally since at least the 26th century BCE. Phylogenetic analyses seem to indicate that the plant originated somewhere in Central Asia, possibly in and around Kazakhstan. And the plant is most closely related to hops that we use to bitter our beer. The phytocannabinoids come from the female cannabis flower trichomes, and there are at least 120 different cannabinoids present in the plant. Here we see a graphic from a recent TIPS article showing all of the different potential ways where there is low quality evidence to suggest that different cannabinoids might act. So all of these arrows really indicate one or two preclinical studies of low quality evidence to suggest different functions that cannabinoids might have in the body. The important thing I want you to take away from this, though, is that in cannabis, the two major cannabinoids that are most abundant are THC, which is a partial agonist of CB1, so it partially activates, and CBD. And CBD does not turn on CB1. CBD acts on many, many different receptors, and CBD does not cause intoxication or the high that we associate with THC. To dispel another myth, there's no genetic difference that's been consistently shown between cannabis indica and cannabis sativa. For all real scientific purposes, these are genetically the same plant, and we should really be thinking more along the lines of cultivars or chemovars that express different ratios of cannabinoids. Cannabis also contains terpenes. We don't know whether or not these affect the pharmacodynamics of cannabinoids. They might affect the pharmacokinetics, and they might also influence a person's experience with drugs. I like to liken this somewhat to different wines in a wine store. So you might go in and taste many different flavors and perceive many different notes in a wine. But at the end of the day, the thing that's going to cause intoxication in the wine is the ethanol. One of the major questions ongoing in cannabis research is this idea of an entourage effect, which asks the question, does the complex milieu of cannabinoids present in the plant influence the pharmacology in a human being? The simple answer to this right now is that we don't know. It seems like many of these things can influence one another at the level of pharmacokinetics. But one of the questions we have to ask around this is, would that be beneficial or harmful? And how consistent would it be from plant to plant? So now I'm going to transition again and start talking about some new information coming out of kind of the latest and greatest research. And I transition with this beautiful image. So this is actually a trichome from a female cannabis plant. This is up here where the cannabinoids are manufactured by the plant. And this image was taken on the Canadian light source synchrotron. And I just think it's really lovely. So when we consider cannabis research, one of the things that we need to consider is that interest in cannabis and cannabis-based medicines is growing extraordinarily rapidly. And I like to put up this operation image, not because I endorse it in any way, but it shows kind of the rapid extent of growth to which people have tried to determine where and when cannabis is useful. However, studies in the past that have looked at cannabis-based medicines have suffered from a few key things, simply because of the difficulty associated with conducting cannabis research. We tend to see issues with blinding and power, as well as heterogeneity of disease. So for example, different modalities of pain or route of administration across studies. So think oral injection of cannabinoids versus smoked inhalation of cannabinoids. I would say studies within the last 2 years have gotten a whole lot better at this. And it's been very encouraging to me and my colleagues to see individuals really step up and try to advance the science in such a meaningful way. One of the areas where we see cannabis research being intensively examined is pain. So to date, there have been more than 100 randomized control trials conducted for cannabinoids in pain. And these are every type of pain modality that you might imagine. Based on meta-analyses, there's some uncertainty about whether cannabinoids improve pain, but if they do, it seems like neuropathic pain is the area where benefit is most likely observed. and the benefit tends to be about a 30% reduction in pain. These are all working, by the way, through that same mechanism of action that I described earlier in the spinal column, where endogenous cannabinoids act to inhibit the release of nociceptive neurotransmitters from neurons traveling up the spinal column. And so there's less of a perception of pain or noxious stimuli. In epilepsy, Dr. Davinsky spoke on this earlier this week. But presently, there have been, 6 placebo-controlled trials of CBD conducted to date. Many of them have been conducted by GW Pharma. And in general, we see a pretty consistent effect of cannabidiol working to reduce seizure frequency, including in disease states such as Lennox-Gastaut and Dravet that are otherwise refractory to treatment. There are side effects that tend to occur, but they tend to be fairly mild, so tiredness, diarrhea, or reduced appetite. In terms of mechanism of action, it's been a real struggle to determine in the body how a promiscuous drug like cannabidiol that binds many receptors is having its anticonvulsant effect. And then last, traumatic brain injury is one that's been increasingly investigated for cannabinoid-based research or cannabinoid-based medicines. And this is something my own lab has been interested in as well, which is why I include it here. So somehow in a field with 27 papers on cannabinoids and TBI, 12 of those 27, at least when I last checked, were review articles. There's epidemiological evidence to suggest that reported use, or 70% of individuals report use prior to TBI, and many people report use of cannabis for recreational purposes or to reduce anxiety or to improve sleep. But from a mechanistic standpoint, cannabinoids do seem to play a role in terms of helping with the TBI process. So cannabinoids being anti-inflammatory and anxiolytic may help in an individual's recovery, at least from an epidemiological standpoint, but preclinical evidence is somewhat lacking. So now I want to talk about the research going on in my lab, and I've tried to sow the seeds throughout what I've talked about so far in terms of what we're doing in the lab as kind of a foreshadowing. So these are some of the people that work in the lab. The picture is a little bit out of date because this was back when we could travel in late 2019. But these folks get the wonderful job of doing the work in the lab, and I get to present that work to you. So one of the projects we have going on in the lab is exploring synthetic cannabinoid receptor agonists. So these synthetic ligands, like I mentioned earlier, they were originally designed as tool compounds to study the endocannabinoid system. They are full CB1 agonists, whereas THC is only a partial agonist. These compounds will fully activate the receptor. They activate both CB1 and CB2, but it's thought that the majority of their human or in vivo effects occur via CB1. In Canada, these are considered Schedule II drugs, and they're also illegal for possession. And in human beings that use these substances, we see tachycardia, vomiting, often violent behavior arising or mania, some suicidality, as well as psychoses following acute treatment, and there have been several instances where individuals have died of overdose. So these are not THC, they are fundamentally different. One of the things that we were interested in exploring as a lab with regard to synthetic cannabinoids was this idea of bias. So at a G protein coupled receptor, different ligands can bind the receptor to force that receptor to adopt various conformations. And here in this infographic from a colleague, we see that each ligand forces a slightly different conformation of its receptor, and the result is that different pathways are selectively activated. We became interested in this because it was thought that if there were certain pathways that were beneficial, that we could produce ligands that would benefit the body more than other ligands whose signaling profile produced more adverse consequences, all through the same receptor. So how does this apply to synthetic spice cannabinoids? Well, we began by screening our cannabinoids at 2 disparate signaling pathways. One was the inhibition of cyclic AMP, which is mediated by the G protein. And this is thought to be beneficial to that normal homeostatic process of inhibiting neurotransmitter release from the neuron. The other was beta-arrestin recruitment. Beta-arrestin serves to scaffold onto the receptor and recycle it, bringing it inside the cell for degradation. So on the one hand, we have cyclic AMP inhibition, which simply put is going to be, good or homeostatic, and beta-arrestin recruitment, which causes your available pool of receptor to decrease over time. And what we saw was that most of the spice cannabinoids were pretty good, as expected, full agonists of the G protein pathway. But in some cases for beta-arrestin, we had select spice compounds such as 3-chloroabpinica that were what I would call supra-physiological agonists, producing 300% or threefold greater responses than our reference agonists. And we thought that was pretty concerning or interesting. Asher Brandt in my lab ran a bias analysis to compare the signaling effects of the synthetic cannabinoids with others. And what we saw was that there were certain of these cannabinoids that were highly biased for the beta-arrestin pathway. What we then wanted to know using the crystal structure for the CB1 receptor was whether or not the actual mode of binding for those ligands was fundamentally different from their G protein biased cousins. And what we were able to show was that, in fact, depending on which ligand you bound to the receptor, so here we see a cyclic AMP biased ligand and here a beta-arrestin biased ligand, binding to that same receptor, their conformations of binding are fundamentally different, and the free energy associated with that binding is also different as well, suggesting that these ligands do in fact adopt different conformations to force the receptor to do different things. That's all well and good, but this is all molecular. What about in a living organism? So to test that out, what we next did was we picked our most cyclic AMP biased ligand and our most beta-arrestin biased ligand, and we ran them through a series of simple behavioral assays in our mice. The first series of assays we did is called the tetrad. And in the tetrad, we test mice for catalepsy or freezing behavior following drug treatment, body temperature, and nociceptive potential. I'm going to work through this backwards. What you can see is that in terms of nociceptive potential, all ligands were very much equal in terms of their efficacy to reduce painful stimuli. However, our beta-arrestin biased ligand was far, far greater at producing a hypothermic response in these mice. You can see a profound drop in body temperature, as well as a catatonia or a catalepsy in these animals that was not seen with the other ligands. We also ran these compounds in the elevated plus maze for these animals and saw that the G protein biased ligand reduced anxiety in these animals, whereas again, the beta-arrestin biased ligand produced profound freezing and reduced movement for those animals that were treated with that drug. So what we saw was that beta-arrestin 2 bias was correlated with catalepsy and hypothermia, but it was not correlated with the nociceptive potential or the anxiolytic effect of activating CB1. So from there, the next place we wanted to explore was this idea of positive allosteric modulation. So we feel that at this point we've established direct supraphysiological activation of CB1, probably not a great idea. But what if we could just use a dimmer switch approach and advance endogenous signaling at the receptor? So to do this, we use what's called a positive allosteric modulator. A positive allosteric modulator, or a PAM, is going to bind some other site on the receptor and augment that receptor's ability to signal through its endogenous ligands. In theory, this is good because you have reduced likelihood of dependence, tolerance, and adverse effects, because your PAM is only active in the presence of your endogenous ligand. We started with this compound, GAT211. Proof of principle showed that it would slow the off rate of the orthosteric ligand from the receptor. However, GAT211 was racemic, as I'm highlighting with this red asterisk. It has a chiral center, if we remember back to our OChem days. And when we ran GAT211 in conjunction with an orthosteric ligand, we saw that although we got an augmentation of potency, we also had a baseline increase in the activity of the receptor, even in the absence of an orthosteric ligand, which suggests that GAT211 was activating the receptor all on its own. So what we did was we separated out the enantiomers, and we were able to show that the R enantiomer of the drug, GAT228, was the thing that was causing this partial agonism, whereas the S enantiomer, which we call GAT229, potentiated both the potency and the efficacy of the ligand, but didn't activate the receptor itself. Therefore, GAT229 was our true PAM. And these are just the bias factors indicating that we had selected for a compound that preferentially activated the G protein pathway. We've gone on since then, and we've been building and developing new, better versions of these CB1 PAMs. We've been able to develop a library of CB1 PAMs that decrease intraocular pressure in an animal model of glaucoma, as well as decrease pain perception in an animal model of pain, which I'll talk about in a second. We've managed to increase the potency of these ligands, so they're now low nanomolar potency, as shown by this graph here, but they're only still weakly active in the beta-arrestin assay, meaning we've managed to retain that bias for G protein. These now are even newer versions of these drugs. So we're now into the 3rd generation development of these drugs, and we've been able to add different subgroups onto the compounds to retain a G protein bias ligand that now has better drug-like properties. So here's some of those data for the pain model, and what we were able to show was that these compounds retain their ability to reduce nociceptive stimuli, which is wonderful. And in a complete Freud's adjuvant model of pain in a rat, what we were able to show is that these compounds are able to increase load bearing on a paw that has a painful stimuli applied to it, suggesting less pain perception, and that they're able to do that in a dose-dependent manner. However, if we run the same drug in a CB1 knockout animal, the effect goes away, showing that these effects are truly CB1 dependent. The next place we've begun exploring the effects of these drugs is in absence epilepsy. So absence epilepsy is a state of epilepsy caused by generalized seizures, common in children who appear to be daydreaming. So there's not a grand mal seizure phenotype, but the children will appear listless or appear to be daydreaming. Individuals who suffer from absence epilepsy also experience learning deficits and anxiety. And the existing treatments, such as ethosuximide and valproic acid, have a lot of severe adverse effects and are only effective in about half of all patients. So for this, we use an animal model called the GARES, or genetic absence epilepsy rats from Strasbourg. These have a spontaneous mutation in their calcium channels, which causes them to have absence seizures. Here we can see, if this works, a video of that. Oh, it's going to work. So here you can see the animal listing off to one side, and it's experiencing an absence seizure, which we then record through an EEG device, which is capped onto the animal's head. At the end of the video, the animal will spark up again, and you'll see it kind of regain its orientation. Oh, sorry. So then at the end of the video, the animal would regain its orientation. So we started testing these GARES animals to see what their endocannabinoid system looked like. And the first place to start was just to see, is their abundance of CB1 fundamentally different compared to wild type? So we call our wild types non-epileptic controls, or NECs. And what you can see is that in the cortex of these animals, there's a profound reduction in CB1 levels in the cortex and the GARES compared to the NEC animals. This difference seems to be specific to the cortex compared to the thalamus, with a slight difference in the hippocampus as well. This is important because the primary area affected by absence seizures in this model and in people is indeed the cortex. We also saw profound differences in 2-arachidomal glycerol levels throughout the brain, but most notably in that cortex again, showing that the endocannabinoid tone was elevated, but CB1 levels were down. We tested our compound GAT229 in this animal model, And what we were able to observe was a dose-dependent decrease in seizure duration and seizure incidence for both male and female animals following either 1 or 2 hours of recording. And we were able to follow this up by directly infusing GAT229 into the cortex, but blocking the effect of GAT229 with some systemic injection of a CB1 antagonist, SR14, to show that this is indeed a CB1 dependent effect. So here in this lovely infographic that a PhD student of ours made for us, we see that in the normal state, you have CB1 receptors working normally, EEG activity looks fine. However, you enter a state in the Gare's animal where there are fewer CB1 receptors and more 2AG hanging around, and you have this elevated absence epilepsy phenotype. GAT229 then is able to come in and take the remaining CB1 receptors and really turn their activity up in order to diminish that seizure response or phenotype. All right, so now I'm going to switch gears a little bit and talk about phytocannabinoid work going on in the lab. And I'd like to put these transition slides in to cue it up for myself, and then we'll wrap up. So phytocannabinoids are present in many, many shapes and forms. There are more than 120 present in the plant, as I indicated earlier. However, Health Canada only regulates content for THC and CBD in cannabinoid products. Historically, we don't have a lot of data on these compounds because they're very similar in structure and lipophilicity, so they're real hard to isolate. They're present in very, very small amounts. And they're still controlled substances, so you still have to go through all the bureaucracy of obtaining the things. Using the same toolkit of assays that I described for the synthetic cannabinoids, we began to explore some of the phytocannabinoids in animal models and in cultured cells. So in this cell culture experiment, we compared efficacy at the G protein pathway as well as at beta-arrestin signaling. And what you can see is that the majority of the phytocannabinoids that we tested were weak partial agonists of CB1 activation through the G protein. Interestingly, though, THC stood out as being the only phytocannabinoid we tested that could recruit beta-arrestin. So it was unique in that way at CB1. At CB2, we saw that, again, the majority of compounds were partial agonists or full agonists of CB2-mediated G protein signaling, suggesting that a number of these compounds may have anti-inflammatory potential. Interestingly, too, at CB2, a number of the compounds were able to activate beta-arrestin 2 signaling, whereas this was not seen for CB1. And this work was recently published by my PhD student, Ayat, in Scientific Reports, if you're interested in having a more detailed look at this work. We tested all of these compounds in that same tetrad mouse model that I described earlier, and we saw that THC, as expected, produced profound cataleptic responses, hypothermic responses, and antinociceptive responses, as well as slowing down locomotion and decreasing anxiety in the open field. However, we also saw some other interesting things, notably THCA, the acid form of THC present in the plant, as well as THCV produced dose-dependent antinociceptive effects in the tail flick assay. THCV also produced a limited catalepsy and hypothermia at high doses. In the open field, we observed that THCA, THCV, and cannabichromene all reduced movement. And for cannabichromene, we've done follow-up studies now, and we don't actually think that cannabichromene hypolocomotive effects are CB1 dependent. It looks like it might be happening through a serotonin receptor, which is cool too. Cannabigerol showed an anxiolytic effect at high doses, and again, we don't think this is CB1 dependent. It might be through a different receptor. Because it's interesting to us and many others, we've begun exploring some of the terpenoid effects. And for some of the terpenes we've screened, we've seen that specifically for curcumin, EGCG, and quercetin, all terpenoids found in many plants, including cannabis, they're able to partially reverse the effects of THC. These effects appear to be lipid dependent and not specific to CB1. So there's something else that we're fiddling with when we provide the terpenes to the cells. So we're following this up now in animals. We've also begun exploring the potential effects of cannabis in animal models using a smoke inhalation model. Historically, the majority of research that was done on animals focused on injection of cannabinoids, but really for the most part, people don't inject cannabis, at least not to my knowledge. So more clinically relevant or translational animal models were sorely needed. And so we set up this smoke-based system in our lab, and we're able to validate the pharmacokinetics for dosing of animals with smoke in a paper last year looking at that same Gare's animal model of absence epilepsy. What we saw was that in this Gare's model, when THC was injected, there was a dose-dependent increase in seizure incidence and seizure duration. Similarly, when CBD was injected, there was a dose-dependent decrease in seizure duration, supporting what's been seen previously with cannabidiol. When we used cannabis smoke, we found that our THC-heavy cannabis smoke, Mohawk, was able to increase seizure duration, and this was not fully blocked by a CB1 antagonist. The CBD heavy smoke that we used, called Treasure Island, had no effect on seizure incidence or seizure duration, suggesting that in this model, THC appears to be pro-convulsant, whereas CBD either attenuates seizure or has no effect. And finally, we looked at the effects of THC on traumatic brain injury. So our model of traumatic brain injury uses a weight drop. In this model, a 300-gram weight is dropped from 1 meter onto an animal, and that animal is allowed to fall through the platform. We did CT scanning on our rats to show that this does not cause a fracture of the skull, but it does cause profound injury in terms of concussive-like or TBI-like symptoms. Following this injury, we treated the animals with a high dose of THC, and then we tracked their performance on the Rotarod task. And what we saw was that animals treated with THC that did not receive a TBI did worse on the Rotarod, specifically male animals. However, TBI plus THC didn't make the animals any better or worse. So TBI plus THC was neutral, although TBI, as expected, did reduce Rotarod performance. The females were a lot more robust to this effect. The females following injury did not show a significant reduction in Rotarod performance, and THC had no effect in the female animals on Rotarod performance, suggesting a higher degree of resiliency and an important caveat with regard to sex differences in males versus females. On the flip side, when we looked at CB1 levels in the cortex of these animals, there was no change in the males. But in females, CB1 receptor levels increased following THC treatment, but were then normalized following THC plus TBI difference, suggesting that in females, CB1 levels are more plastic or more adjustable over the course of treatment or injury. And those studies are ongoing as well. So we have a bunch of other fun stuff going on in the lab, but I can't talk about it all and for the sake of time. As you can tell, though, I love talking about this stuff. So if you'd like to chat with me more, let me know. Excuse me, we're looking at things like the endocannabinoid system in pregnancy in collaboration with Dr. Howland at my institution. One of my PhD students is exploring interactions between the endocannabinoids and orexins in terms of appetite and sleep. I have a program and a student exploring specifically PAMs for CB2 and new compounds for an orphan cannabinoid receptor, GPR55. We're also developing PET probes with some of our compounds with a colleague in chemistry. And I have a new and growing program exploring similar approaches to design ligands for the serotonin 2A receptor, led by my PhD student, Asher Brandt. So there's a lot of research in this young and growing field of cannabinoid science, and there's a ton of interest. Cannabinoids might be useful as medicines for some indications, but high quality evidence remains sparse. CB1 has this really unique allosteric site that we think we can target to augment endogenous cannabinoid signaling while avoiding intoxicating effects. And all of our data collected to date seem to suggest that really the way to go is to assess G protein bias at this receptor. less abundant phytocannabinoids do appear to have effects on our body's endogenous cannabinoid system. And I think as a drug discovery person myself, even if we don't find that these having effects within the plant, we could develop new drug scaffolds based on the compounds that we find. THC is unique because it seems to be this phytocannabinoid that uniquely activates beta-arrestin 2, and it seems to reduce coordination in male TBI rats, as well as exacerbate seizures in the Gares model. So it's all connected. Increasing evidence shows separation between G protein and beta-arrestin signaling, and we want to explore this further. And that's where a lot of my research is going to go, is exploring this difference in ligand bias. As we develop our CB1 PAMs, we're increasingly interested in the pharmacokinetics and developing better drug-like properties for these drugs, as well as assessing the pharmacology of cannabinoids at other receptors, orexin receptors, serotonin receptors, orphan receptors. And we're going to continue to assess the translatability of our findings into animal models like seizure and TBI and pregnancy as the ones we're focusing on. So science is a team sport. Thank you so much for your attention. All of these people on this slide, as well as these many funding agencies, have very generously supported our lab. I'm very grateful to all of them. And I'm very grateful to you all for listening to me. Thanks.
Thank you very much, Dr. LaPrairie. That was very interesting. Definitely brought me back to my second year organic chemistry, especially at the beginning. So we'll open it up for questions now. If there are any, please type them into the Q&A box or into the chat. Can you talk a little bit about the drug safety piece that you mentioned right at the very end, as we're waiting for people to put into the question box?
Yeah, for sure. So one of the things that we're interested in my lab is not just, seeing where we can develop new and great drugs, but recognizing that all drugs have limitations. So now with any new drug that we develop, we're not just assessing the beneficial effects, but we're looking at pharmacokinetic effects in terms of drug disposition and metabolism, as well as potential toxicities on cardiac health, locomotion, and behavior, for example.
And with the fact that CB receptors are so prolific across the whole body, is it difficult to get drugs to act in certain ways?
Yeah, it's very difficult. My hope is that, kind of being able to focus in on this idea of bias and select for certain signaling pathways, we might be able to limit adverse effects. I don't think we'll ever have like this perfect wonder clean drug, but, make things better at least.
All right. Question from Elizabeth. Is there any evidence that changes in CB2 receptor density or receptor number due to peripheral injury can influence CB1 receptor density in the brain?
So I believe there were some studies done, not with regard to injury, but in the context of infection and also in the context of addiction biology. These were done by NIDA in kind of the mid-teens, like 2014, 2015, that looked at regulation of CB2 in the periphery and were able to show that changes in the inflammatory state or CB2 in the periphery would influence the endocannabinoid system centrally. I don't know that work has ever been followed up on though. So there's some limited evidence from animal models.
All right, a question from Eliane. To what extent do you think the actions of cannabinoid compounds depend on the underlying genetics of the animal? For example, the effects on the seizure model with the defect in the calcium channel?
Yeah, that's a great question. So it's certainly possible that, for example, the underlying genetics of our animals could influence the efficacy of our drugs. And in fact, most of my studies are run in C57 Black 6 mice. And the reason we use those mice is because they show a larger cannabinoid dependent effect than, say, a bulb C mice. for example. So genetics definitely plays a role. One of the things we started doing with all our new drugs is before we test them in animals, I send them away for a safety panel that screens the drug for 75 different receptor targets. So for example, screening against beta receptors in the heart, and calcium channels are in that panel as well. So that safety panel helps us to rule out things like the calcium channels. And I know that for the GAT compounds, for example, they don't act at that calcium channel. So we're pretty confident based on that safety screen that we can rule some of those things out.
Great. From Julianne, fascinating observations in mice. Do you have any idea how far away we are from making observations in human trials?
So for the synthetic cannabinoids that I talked about, the PAMs, we're probably 5 to 10 years out for those. So those will be a while. For the plant-based cannabinoids, I think we're already starting to see growing interest in testing those cannabinoids as purified substances in humans. I know of a few individuals even in Canada that are interested in doing small-scale human clinical trials to look at, say, cannabichromene or cannabigerol for certain disease indications. So I think we'll see those phytocannabinoid data within the next couple of years.
Great. Jason, do you have any final comments or questions?
So I just had to get my video working. So thank you for an excellent presentation. I have lots of questions that stem from it. I'm very curious about the what will be the clinical applications of this. It seems like, despite the fact there's been decades of research into cannabis, there's an enormous amount that we still don't understand about potential therapeutic applications. I'm particularly excited about the idea about, targeting new molecules that are going to actually maximize the benefit, reduce the adverse effects. Fascinating to sort of see these different trials where you can sort of explore with very similar components, but how their particular actions take place within these animal models, and ultimately what's going to be human trials. One of the challenges that we've had in Canada, as I'm sure you're aware, is the requirement by the regulatory agencies for products that we use in clinical trials to be GMP compliant. And I mean, your work is going to get around that. I mean, because you can actually produce these things in the same way that typical pharmaceuticals are. So there's tremendous appeal for that, being able to sort of parse out the beneficial aspects of this complex plant and take away a lot of the sort of noise from it and maybe some of the components that are causing harm. So I think your work is definitely important to watch. And ideally, we're going to be able to get some of these components out into clinical trials and then extend your work to see what the impacts are. So I really thank you for your time today, your presentation.
Thank you.
I learned a lot, and I'm sure that the rest of the audience did as well. And we look forward to having you back in future conferences so that we can hear about the next steps and the next discoveries that you and your lab are coming up with. So again, thank you very much for your time and your presentation, and really appreciate it.
Yeah, my pleasure. Thank you so much for inviting me. This was wonderful. I always enjoy talking with you guys. Thanks so much.
Fantastic.